Hi, thank you for inviting me to present my poster and what we achieved so far. My name is René Lenz and I'm from the University of Applied Sciences FH Campus Wien in Vienna. This work was done in cooperation with my supervisor Alexandra Graf, also from the FH Campus Wien in Vienna. The title of this work is Comparative Analysis of Urban Microbiomes Using Machine Learning Algorithms. Several recent metagenome studies deal with the microbial composition of public spaces in urban areas. In particular, public transport systems are addressed and samples are taken from different surfaces such as handholds, seats, or the passenger's palms. There are approaches to investigate the differences in the functional composition of the samples. Others focus on the differences of antibiotic resistances. Another approach is to differentiate between different cities based on the abundance of species present in the samples. These studies were implemented with random forests as well as support vector machines and multi-layer perception. The most promising results were achieved with random forests so far. The focus of this work is on finding key species in order to differentiate between different locations with the use of random forests. For this purpose, whole genome sequencing data from the MetaSub consortium provided by CAMDA was analyzed. The raw sequences were quality checked with FastQC and Tremomatic, and then human reads were removed by mapping the reads against a reference genome with bowtie. The remaining reads are taxonomically assigned with Kraken and Braken. Ecological assignment then was done with BugDive database, and so the further gathered abundance files were then analyzed using R. The data is first visualized, evaluated, and further a random forest analysis is used to try to reassign the samples on different cities. The data set was kept small and currently includes the following cities: Singapore, Vienna, Ilorin, and Kiev. It was also tested whether it makes a difference if you keep only those species that are present in only one of the cities at a time, or if it is better to keep all the species. Our approach was to study the top abundant species of bacteria. Therefore, we used top 10, top 20, top 30, and top 100 species of each city based on their abundance. The PCA plots provide a first insight to visually detect if there is a certain separation of the individual cities or not. There's no clear separation visible, but a certain division can be seen, at least if you look at the top 10, top 20, and 30. With the top 100 species, the distribution seems to be a bit more homogeneous. With the same data, we then did a random forest analysis. The class error of the individual cities in the random forest analysis predictions indicates that it makes a difference whether one uses only those species that occur in only one city at a time, or whether all species included are also included into the analysis. 10 times the class error of the respective city has decreased, including all species. 1 time it has increased, and 5 times it remained the same. Also, surprisingly, within the top 10 species, several were involved in bioremediation, like degradation of heavy metals, the degradation of industrial dyes, toxic pollutants, or hormones. Other groups are clearly plant-associated, often involved in nitrogen fixing or part of the human microbiome. This leads to the hypothesis that the level of pollution might play a role in the classification of the sample origins. Further considering the top 20, this selection is complemented with several more human microbiome associated species, as well as with some water species. the fact that the random forest seems to perform better if one considers all the species, not just those appearing in only one city, leads to the assumption that there's not simply a special set of key species for each city, but that there is a complex relationship between species and abundance, and certainly a lot of other metadata. To support these results, it is necessary to include even more data into the analysis and to optimize the parameters of the analysis. It is also necessary to use some other analytical methods to compare the results. Another point with projects that take place in different places all over the globe is to ensure that the sampling and the processing of the samples is done in the same way to get a balanced and comparable data set. Yeah, and with this basis, such investigation could provide even more accurate results. And this now brings me to the end of my presentation. I would like to thank you very much for your attention. And if you have any questions, don't hesitate to contact me or Alexandra Graf.